Hello, hi. I'm Dr. Dave Ories in New York City. It's November 14, 2020, and today I'm talking about the Pfizer vaccine for COVID-19. I've got a lot of questions about that. So I was going to explain a few things about the difference between a vaccine like a flu or measles vaccine and this new vaccine for COVID that Pfizer is testing and had some preliminary positive results, which there's more testing to do for a few more months. But the questions I get is, what is an RNA vaccine as opposed to a regular vaccine? To explain that, I explain a regular vaccine. How that works is, what you do is you take the virus particle has antigens on its surface. An antigen are little proteins, like you'd see like little thumbtacks on a basketball. So they're tiny little things on the surface. How a vaccine works in general is you take one of those thumbtacks, which is a protein, very tiny, and you make a lot of them in a lab. And that's complicated, but that doesn't matter right now. Then you inject that in someone's arm, like for the flu, for example, and what happens is your B cells or your white cells will notice that, and they will grab onto it and make what are called antibodies against that protein. So now the B cell has been educated, if you will. So 2 years later or months later, whenever, depending on the vaccine, if you get exposed to that virus, measles, polio, whatever, then your B cells have a memory and they recognize this antigen right away and make millions and millions of antibodies very quickly, within an hour or two. And those antibodies latch on to that surface protein on this particle, rendering it harmless. That's how vaccines work, or reducing the number of particles. So instead of with the flu vaccine, you'll still get the flu, you might get sick, but you'll have a 90% chance of being much less sick or not sick at all. So basically, a basic vaccine is you manufacture these surface proteins and then you inject them into someone's arm and they make an inflammatory response, and you educate a bunch of B cells that are now ready to fight this virus in the future. The Pfizer proposal, which is a different approach, but the end result is the same. The idea is to end up with the surface protein, the antigen, in your bloodstream so the B cells can find it, recognize it, and make antibodies. Now, how the protein gets there with the Pfizer vaccine, that's what's different. What this does is a little more tricky, and I'll explain. When a cell makes a protein, this is kind of as simple, I think, as I can make it. Cells make proteins and other things. They're like manufacturing entities. So an example is a pancreas cell makes insulin. Well, how does it make insulin? Well, insulin's, let's say to keep it simpler, say insulin is a chain of amino acids, which is a protein. But it has to be exact. So it's 1 through 10. It's 10 different amino acids, say. Insulin's more complicated, but it's good enough to understand the metaphor. So it's going to be a protein, which is a chain of 10 amino acids, which are little molecules. So 10 in a row. A very precise order. It has to be precise. So what happens is, in the nucleus of the cell, a little piece of DNA opens up, and then what's called RNA, which is a ribonucleic acid, but it's not important, a different kind of molecule, that makes a copy of that piece that opened up. But it's the opposite copy. So A becomes T, I think G becomes C, whatever the letters are, doesn't matter. It's an opposite copy, a mirror copy. That copy of the piece of DNA floats out of the nucleus of the cell, the middle bit, and goes in what's called the cytoplasm, out in the wider, bigger part of the cell. Still inside the cell, but not in the nucleus anymore. That little piece now, what happens is, called transfer RNA, these other little molecules, they latch onto it. So this is called the DNA code. So let's say, imaginary, the letters ATT means alanine. That's the code. It's that simple. So there's 20 or so amino acids. Each one has a 3-letter code, which is unique. So if I have a chain of 30 letters, 3333, that will translate into an amino acid of 10 amino acids. So let's say that again. If I have a chain of 30 letters, A, T, T, G, C, T, T, G, T, whatever, 30 of them, then the transfer RNA will land on that and that's like 3, 10 times, and you'll get, on the other end of that, it will create a chain of a 10 amino acid protein. Then that will float away into the cytoplasm. Then it does it again and again and again and again. So that one little piece of messenger RNA will manufacture thousands of insulin molecules. Now, that's called amplification. So one piece came out, but doesn't make one molecule, it makes thousands. Now, further complicating this, or further you should know, is that when that piece of DNA opens in the nucleus, it doesn't get copied once. It gets copied hundreds of times. So you get hundreds of messenger RNAs in the cytoplasm. So let's say you have 100 of those. Each one makes thousands of copies. That's all of a sudden you get hundreds of thousands of molecules of insulin. That's all called amplification. So they call it that because a single piece of DNA that opened up in the nucleus to make insulin will be responsible to make millions of copies once it gets in the cytoplasm. Those that are released into your bloodstream, and you have insulin. That's how it works. Of course, the pancreas has millions of cells, so you see how quickly the numbers get very big. And you can make, cups and cups of insulin. Not cups, but, milligrams or milliliters, whatever, a lot of insulin. So why did I tell you all that? Because this is how the Pfizer vaccine works. They've created a little tiny nano-sized little container, call it, that they inject in the muscle of your arm. Those little tiny containers, which are smaller than microscopic, incredibly small, each one contains thousands of these messenger RNAs. So that will go into your muscle cells. The cell brings it in. It gets into the actual cell, but not in the nucleus, just in the cell cytoplasm, the public area. And then this little capsule opens and releases a bunch of messenger RNAs. Now, that cytoplasm will start making proteins. And in this case, the protein won't be insulin. It'll be the antigen that you want. So it's a sequence of amino acids all connected together. It's slightly more complicated, but this is the basics of it. And it will start manufacturing thousands and thousands and thousands and thousands of these proteins, which is the antigen that you want. That will float out out of the cell, into your bloodstream, and your B cells will see it, make a reaction, and make antibodies against that antigen. It's very specific. It's incredibly specific. Imagine a key with 50,000 teeth. It's that specific. So the antigen has to be exactly the antigen. It can't be close or kind of. So now you have that protein, the antigen they call it, floating around your blood system, and your B cells see this, and then they manufacture antibodies. And they also remember forever, or months or years, depending on the situation, the vaccine, this particular antigen. So if they see it 5 years later, boom, they instantly make antigens in days, as opposed to the first time you got the shot, which would take 6-8 weeks before you have antigens. So if you get infected with this virus in the future, you have an instant response, as opposed to response 6 weeks later when it's too late. So I hope that makes some kind of sense. You can text me, email me. I'll be more specific, answer any questions you have. I'm going to do a weekly live, Dr. Dave Live, once a week. I don't know when. I'm guessing, Wednesdays or Thursdays. I'll figure it out. And I'll answer any questions anyone has. So the end result is the same. When you inject antigens in someone to make a response, you're just injecting the proteins directly. They make an antibody response, and in days or so, those proteins go away. They break up. The difference is with the Pfizer situation, these muscle cells are making antigen, COVID antigen, maybe for days or weeks. So continuously all the time, 24/7. So you can have a lot more of these antigen proteins in your blood that will allow the B cells to make a stronger and stronger and stronger response. You get more and more B cells floating around. You're talking instead of 1,000, 100,000, or 1,000,000. There's large numbers with these things because they're very tiny. People ask, well, can I catch the virus? Will I get COVID? No, you can't catch COVID because it's only a single tiny piece of the virus. If you use a car as a metaphor, this would be like the left door handle. So you can't make a car. If I gave you 10,000 left door handles, you can't assemble that into a car. Same with the virus. It's a little tiny protein, which is one, 10,000th or 100,000th of the parts you need to make the whole virus. So there's no virus in the vaccine. The other concern is about this RNA. Are you manipulating RNA? Well, yeah, you are. You're tricking the cell into making the protein you want it to make, which will be this antigen. How can that go wrong? Well, maybe it could go wrong if it never turns off, but my understanding is that this M messenger RNA that they put in the cell doesn't last that long, and it makes days, it works for days, and then it will kind of break apart and not be there anymore. And by that point, you have many, many thousands and thousands of B cells, which are now immunized. So I hope that helps explain some of this technology. And any questions, you can email or message me, and I'll be happy to answer anyone's questions. So this is Dr. David Ories in New York City. That's my COVID vaccine story. Take care and good night.